expertise and support. Just a small disclaimer, whilst Carbex definitely supports these activities, and the content of this is supported by Carbex. Carbex's funding is sponsored by the Cooperative Agreement Number ID SEP 160030 from ASPR BARDA, and by awards from the Wellcome Trust, Germany's Federal Ministry of Education and Research, the UK Global Antimicrobial Resistance Innovation Fund, and the Bill and Melinda Gates Foundation. The content is solely the responsibility of the authors and does not necessarily represent the official views of Carbex or its funders. With this, I would like to request Taslim Arif Sayed. Dr. Taslim Arif Sayed is the CEO and Director of CCAM. He has had his initial training in neurosciences and received his PhD from the Max Planck Institute of Brain Research in Germany. He did his postdoctoral training in UCSF and also trained in biotech innovation from QB3 at UC Santa Cruz, UC Berkeley, and UC San Francisco. He has been a mentor to many startups and is deeply involved and engaged in promoting innovation, entrepreneurship, and has a particular interest in translational research. With these words, request Taslim to make his comments.
So thank you. Thank you so very much, Narita, and good morning, everyone. I think it's a fantastic, initiative supported by Carbex at CCAM, where we are aiming to build early stage ideas to support much of what one would call it as the efforts in the AMR field per se. I think we are seeing significant participation today, and I would guess that this participation is from across India. And this clearly indicates the interest in researchers and innovators in the field of AMR. It is more significant that this interest is there because AMR as a field impacts largely the whole world, but many countries in the world have a crucial role to play, and India is one of them, where the broad aspects of what one would call it as the impact of AMR has been significantly strong, and the projected numbers are just growing according to what experts say. We have a recent publications on AMR, which has terming AMR as one of the biggest cause of mortality in terms of diseases as we go forward. At the same time, the Lancet paper indicated almost loss of 1.2 million lives per year already. And this number is projected to be what one would call it as around 10 million lives lost per year in 20 to 25 years. And that is a significant number if you're looking at 10 million lives. And collectively, as a community of researchers, scientists, innovators, we need to build solutions to bring those projected numbers down significantly. And the only way we want to do that is to work together and build solutions for preventing AMR, build solutions for early diagnosis and detection of AMR, and then, of course, very importantly, treat AMR as it happens. One of the aspects that we need to look at is to understand the issues, and what at CCAM, what we have been doing for over many years, and as Narita mentioned, has been working with AMR innovators for many years and has been recognized as the possibly most intense platform for building AMR, not only in India, but in a larger region, with a clear sign that we have to go a long way and build collaborations. And we have just built a platform called India AMR Innovation Hub, which attempts to bring together the stakeholders who are interested in working in the field of AMR from multiple perspectives. And this hub is hoping to take some of the converged, as one would call it, the attempts, efforts, and building new efforts to bring down the spread and the impact of AMR that happens in India and beyond. I think today's workshop is another effort to build the community, have experts who are with us to share their thoughts on the field of AMR. We have phenomenal experts like Dr. Bal Ganesh and Dr. Ravikumar Banda in the inaugural session. And what we are hoping to do together with you is to, understand more where we need to work, understand what is required, what are the ways we can build innovations together. And of course, please come back to CCAM as your ideas progress, because while we are bringing everyone together, we work individually one-on-one with innovators to take their ideas forward, starting from funding, from supporting their lab. Next stage, we are supporting the acceleration and many other industry connect, investment connect, and so on. So there are many efforts which continue to happen at CCAM. So I think this workshop is another opportunity for us to discuss the aspects of AMR, the efforts in AMR, the trends in AMR together today, and then follow up with meeting in a smaller groups, meeting on one-on-one, and take some of the thoughts together. But excited to be here, and thanks to the entire team working working on this effort, and hoping that the day brings a lot of exciting dialogues, exciting thoughts, exciting initiatives together, and we all can meet and follow up on that. Thank you from my side, and I'll hand over back to Nanita. Thank you.
Thanks, Tasleem. Just to quickly, before we begin, just to say that, as Tasleem mentioned, we've structured this workshop to take us through the life cycle of an innovation. So the first session, as you all know, will be on will focus on the discovery and the the early research. And as we progress through the day, we will go through the validation, the regulatory, the funding, and the the other aspects as well. So with that, would like to welcome our speakers for the session on early discovery. The workshop focuses on both therapeutics and diagnostics. We have Dr. T.S. Bal Ganesh, kindly mute yourselves and unmute only when you want to ask questions. Thank you. Dr. Bal Ganesh is the president and member of the board of directors at Gangajan Biotechnologies. Prior to Gangajan, Dr. Bal Ganesh was head of research at AstraZeneca, where he led the unit to deliver AstraZeneca's first anti-TB molecule for clinical development. He has extensive work in this area, as well as also led the teams there for the diarrheal disease and broad spectrum antibacterial teams. As a distinguished scientist, prior to his at AstraZeneca, he has mentored several drug discovery programs in various Indian national laboratories under the government's CSIR organization or institute. Welcome, Dr. Bal Ganesh. Our next speaker in the session is Dr. Ravikumar Banda. Dr. Ravikumar also began his career at Astra with Dr. Bal Ganesh, along with, rather with Dr. Bal Ganesh. Dr. Ravikumar is currently the founder of Exciton and functions in the capacity of chairman and managing director. He's a physician turned entrepreneur, or physician scientist turned entrepreneur, and has been instrumental in developing and successfully launching diagnostic products. He and his team at Exciton is credited with the development of the first indigenous ELISA kit for the detection of HIV infection. As I mentioned, he was earlier with Astra, where he has also worked on the research for on Mycobacterium tuberculosis and neurocystic cirrhosis. With that, I would request first Dr. Bal Ganesh to tell us more about the research program at for antibacterials, and then over to that. We will take questions after both the presentations are over. Please use the chat mode. It's easier to moderate questions that way. Just continue to add your questions in the chat mode. We will address them after both the presentations are over. Thank you.
Thanks, Nandita. Can I share my screen? Is that visible?
Yeah, we can see it.
Okay.
You would like to take it on full screen mode?
Yeah, I'm just doing that. Yeah.
Sure. Thanks.
So, good morning, everybody, and thank you, Nandita and Swati, for inviting me to talk in this context. And thanks, Taslim, for I always think that the intensity with which CCAMP is sort of, I don't want to use the word pushing, but creating awareness of AMR is very, very important. And I think it's a great job being done there. So good luck and hope it will continue. So what I'm going to do, that title, AMR Innovations from Discovery to Commercialization, is the title of the workshop. I'm going to talk about the discovery cycle. And I have some favorite slides of mine, which I can't but go through before I come to the discovery cycle. Now, as I was trying to put together a few slides on what do we talk about under discovery, when you say discovery cycle, one of the things I realized is that each topic under the cycle, you could go into depth, and that means each one of them will be a full-fledged talk. And so I've tried to avoid that. I've tried to give an overview, but I've tried to bring in critical, what you would call as critical phrases along the path, which are important to think through right from the beginning, so that you know, tomorrow when you're going for funding or you're trying to explain to somebody what you're doing, there is a mixture of what I know I want to do, as well as you know, the science behind what I'm going to do. And of course, these things keep changing as we go. So going to the next slide, this is one of my favorite slides. This is Voltaire, sometime in 1700, who said, "Doctors pour drugs of which they know little, to cure diseases of which they know less, into human beings of whom they know nothing." All right. Now, this is obviously before you know, a lot of knowledge on each of the medicines we worked out on. But what is happening is that with the explosion of knowledge, with the explosion of information, I should correct it, not necessarily knowledge, information which can be which can lead to you know, expansion of knowledge. It's very humbling. While this exactly may not be true, but if I was to try to treat infection today, the amount of knowledge base that we are developing based on how the microbe interacts with the host, what's happening in the host, sometimes you tend to look at things in a very isolated fashion, and that we need to be aware of. So while this was true some time ago, this has a different context as we go today. And then, you know, this is one of my favorite slides where penicillin and World War II, and we know this advertisement saying, "Thanks to penicillin, the soldier will come home." And this, I think, is in a very big way the start of the antibiotic revolution. Now, this is another slide which gives you an overview of how the whole, you know, this whole bunch of antibiotics have come. And what I want to point out, 60s and before, this was all mostly driven by natural products, synthetic chemistry, serendipitous discovery of new chemicals, but it was all mostly chemistry driven. And in the 2000s, we had, you know, there was a huge gap. And then we had some new molecules coming in. And perhaps the last molecule which came in, if I'm not mistaken, was daptomycin. And we're still struggling to bring new molecules. So one of the things that I always ask myself was, what was happening between 1960s to 2000? This has big relevance in, excuse me. What anyway I'm going to talk about, remember, is drug discovery as applied to infection and not necessarily other areas. So what was happening between 60s and 2000? This has got impact from what we are doing today. So what really happened was what you would call as in this era, 70s to late 90s, it was all chemical evolution. This is just one example of where we started with a gram-positive cephalosporin, and we landed up with a broad spectrum like cefprima, which is a fourth generation cephalosporin. So while it looks like there was nothing happening between 60s and 2000, the whole pharmaceutical industry in infection was being driven by modification of existing molecules, early molecules which had been invented. Okay, so now if you look at, so let's look at this entire period to where we are today. So we started with synthetic, I remember the very first Salvarsan was one of these dye products. Then we had natural antibacterials and antibiotics. Then we had antibacterials through chemical evolution. Then again, there was very big push of synthetic antibacterials. And then all of a sudden, the progression stopped around 2000. Now, there are many factors here. You know, you can say industry was not interested, blah, blah, blah. But one of the things which strikes you is that what happened around early 2000 was this whole business of genome came, you know, everybody said, I'm going to do drug discovery. We had bacterial genomes. And all of a sudden, from 2 or 3 targets, we had 200 targets, 300 targets. So I felt that, oh, wow, this is how discovery should be done in the infection area. And so the whole thing shifted from a chemical revolution to actually a biology-driven revolution. And clearly there has been a big question mark on this success, and we can talk about it, that this whole genome-only based approaches have repeatedly not had high success rates. But today there's a much better rationalization. We tried to use both these methods, multiple approaches, including genome research, synthetic medchem, naturally occurring molecules, repurposing, etc., etc. What's the relevance of this is actually where do you start from? And I'll come to that in a minute. But another one which I wanted to, and this is one of my favorite slides, is if you look at these 4 different events on the left, there is possibility of taking a catch. And so when you launch a drug discovery program, you have to ask, what's the opportunity? And that gets defined into certain aspects, which I will talk about very early on. So ask yourself, what's the opportunity? Then drug discovery is a very, very strong team effort. The baton goes from, that's the lower right panel, goes from, you know, early discovery where it is biology to lots of chemistry to pharmacology to in vivo efficacy, etc. And that baton has to move smoothly. And many times, of course, there is more clarification, but it's a very strong team effort. Then coming to the one on the right upper panel, clearly you have, you know, individual brilliance. Problem solving is a very, very important aspect of drug discovery. And lastly, on the left, speed, right? Many of us come from academics and where we have interesting ideas, and then we want to convert this into a drug discovery program and then take it forward. Drug discovery is a business. If you have a bright idea, you will 99.99% others have also had that idea. And so others are also moving on that. So it is a process where speed is very, very important. And that's why, many times you get, okay, we will review this in 6 months. Means 6 months. It doesn't mean 10 months. So one of the things that you have to constantly keep in mind is how fast am I moving? And this has implications both in terms of, what are the resources you're putting in, what are my milestones, funding, and most importantly, when are you going to apply for a patent and how much lifespan is there for me in the patent? So remember, drug discovery has at least these 3 or 4 facets, which are very, very important as we go forward. All right. Now I'm going to take you through the cycle of drug discovery as far as infection is concerned. And I've tried to, I will deal with early discovery, late discovery, and I'm sure there are other aspects of development, which I think you're going to hear later part of today, including registration. And the first sort of definition, which I put up there, is TPP, and that's the target product profile. And many of us, and what does that TPP mean? TPP simply means, what is my product going to look like? And when you start a program, this looks so silly. I don't even know what my molecule is going to look at the end. How can I actually define a target product profile? Now, if you don't have a TPP, your investor is not going to put in money. He needs to know, what is your unmet medical need? What is the indication? For example, if you are in the infection, what's the unmet medical need today? Just to give you an example, if I say I'm going to come up with another compound which works on Staph aureus, my interest is much lower than if you tell me I'm going to come up with a compound on Pseudomonas. That doesn't mean that Staph aureus compounds are not required, but then where is the current portfolio of drugs or the standard of care? Where is it failing? In what indication? So you got to define that. It may undergo changes as you go forward, and nobody's questioning that. Well, there will be a scientific rationale why that is happening. But today, as a concept, I need to define TPP. And most of us are very reluctant to define TPP. Okay, so now you've got the bug, you've got the indication. Who are your patients? Now, again, the question is, can I define that? Yes, you can. Are you going for hospitalized patients? Or are you going to the community patients? What sort of patients are you looking at? Serious patients, patients who have an early indication, some ideas, mode of treatment. This is all a part of TPP. This may change as you go forward. But clearly, if you ask me today, what's my preferred mode of treatment? Oral. But let's say I'm working on a non-traditional antibiotic, which is a protein. I won't say oral. Then I have a lot of explanations to do, and I have a lot of hurdles to cross, which will make my entire effort unattractive. So I say, okay, I will go systemic, IV. Now, the moment I say I'm going to treat IV, what does it define? It also defines the patients, because I'm going to go hospitalized patients. Then if I'm going to hospitalize patients, then it also impacts my indication. What's my indication? I'm not, for a common cold, I don't go to the hospital. So it's going to be serious indications. So you have a cascade based on each of these definitions. You've got to be aware of what is your comparator. If I come out with a molecule which is just as good as a molecule which is already available, why do you expect an investor at the lowest $100 million to find another molecule? Why is my molecule, what do I envisage my molecule to be superior? How do I differentiate? And that differentiation is important throughout the cycle. You can be an also-ran, nobody's questioning you on that, but for finding an investor, putting in that amount of money for an also-ran project is not going to be easy. What's your competition? For example, if today I went in and want starting a program on a gram-negative bug, there is a lot of competition. There's no question. But it's not a competition where there are 15 other drugs out there which are all cleared by FDA, cleared by various authorities all over the world for the indication that I'm looking for. My competition is way ahead of me. So you need to place yourself from a competition saying, okay, what do I know about it? You've got to do some what we call as, most of you know emotional intelligence. This is competitive intelligence. TPP. So please remember, I know many of you who start drug discovery programs don't want to define a TPP. But the first thing an investor likes to see is what is that TPP? Now, is a TPP written in stone? That's the biggest problem we have. No, it's not written in stone. Obviously, as you progress the project, as you find the new properties for your compound, Nandita, give me a heads up 2-3 minutes before my time.
Yeah, sure, Dr. Madhan.
Thanks, Nandita. All right, so I was talking about TPP. Please spend time defining a TPP. You can change it as you go, but that you have understood where this whole project is going is indicated by your TPP. Then you can call this, many times just to make sure that, I'm not held to exactly what I say in my TPP. Very early on, you can call it an aspirational TPP. Then as you go down to, let's say, late stage discovery, your TPP needs to get really solid. So that's also an evolutionary process, but it's very important. All right, so my second point that I have here is target. Now, first, the question which is thrown to many of us is, has it to be defined? No, today we know we can go to FDA without defining the target. But there are real great advantages if you know the target. One is the whole discovery process itself. You can have good assays. You can look at many molecular pathways, etc. If you know the target. But what does really knowing the target from a FDA perspective, from a regulatory perspective? When you have a new compound which you want to go into man or woman finally, there are, from a safety perspective, from a toxicology perspective, there are two key divisions that we look at from a risk perspective. The first one is called class-specific risk. And the class-specific risk is generally about similar compounds, like if it's a quinolone, right? All quinolones have an inhibitory effect on the bone marrow, irrespective of what modifications you make, fluoroquinolones. Now, that's the class type of risk. The second type of risk is the mechanistic risk. And the mechanistic risk is the target that you are going after in the bacterium, is it also present in the human? If it is present in the human, is the target's function mechanistically similar? If it is mechanistically similar, what will be the effects of inhibiting the target downstream? So it really helps a lot of allaying the fears of the mechanistic risk portion of it if you know what the target is. It doesn't take it away, but it definitely reduces the risk and so the regulatory people get more and more reassured when you know that the target is, or the target's homologue is not present in the human. Of course, there are lots of subdivisions on that, but the second part about target is many times I'm asked the question, knowing in the pathway, is it good enough? I think, yes, starting, but as we go forward, I think it's extremely important that we know what the target is. Personally, I prefer to know the target and I may launch a program, but clearly there are multiple examples where there are components which we don't know the target, or as we get more and more sensitive ways of looking at the mechanism of action, we find the target. So yeah. Of course, what I've not gone into is that how do you start a program? That's why I told you can start it from a chemistry perspective, from a biology perspective, from a repurposing perspective, from, somebody has got a molecule which is patented, published, and you say, okay, I can find a molecule which is different from that, but has the same mechanism. Various ways where you can start a program, but remember about the target. Now,
Dr. Balganesh, about 5 more minutes.
Excellent. I've not even gone to my next slide. Inhibitor. One of the things that we normally find ourselves in trouble is that we focus a lot on the potency. Try to, balance it. Try to, get a chemist to look at, is that compound has properties of a drug? What sort of inhibitor? If you look at all the naturally occurring antibiotics, they're all allosteric inhibitors. The newer synthetic compounds are all competitive inhibitors. Which is better? Just remember that each brings its own positives and negatives, but you should try and know what sort of inhibitor. The next point that I have is FTO. This is extremely important. Now you have an inhibitor, you're progressing the inhibitor from an inhibitor to a drug. You've got to know whether you have freedom to operate. You have to do an early patent search. What are your limitations? Which areas should the chemistry not go? Which areas is the chemistry free to go? And then you try to spend time understanding how does your molecule interact with the target. At this point, you've got a bunch of compounds which you have a decent potency. You answered some of the question of FTO. Now you start working on a variety of properties which needs to be standardized. Of course, I said potency, but please remember physiochemical properties are extremely important. And among that, solubility. If you are struggling with solubility, raise the flag right in the beginning. You need to know MOAs, mechanisms of action. You're starting to looking at efficacy, preliminary efficacy in animal models. You want to know what your spectrum is, so you should be able to test it on hundreds of strains of either Pseudomonas or Staph to make sure that your compound is working along all these strains. You must have an idea of what is your frequency of resistance, because many times certain pathways, certain targets have a very high frequency of resistance, and you want to avoid that. You've got to have some idea of what are the off-target interactions. So this is a panel of usually kinases that you look at, and these targets, we're talking about human targets, to make sure that your compound which inhibits the bacterium doesn't work on other targets and on the human host. And of course, you start safety, like, does it have hemolysis? Is it cytotoxic, etc. When you have defined this, you have an early lead and a good point to apply for grants. I quickly finish the next slide. From an early lead onwards, the project starts to look at in vivo. So we need to understand pharmacokinetics. What is pharmacokinetics? Basically giving so much of the drug to the animal, how much is found in the blood, how long does it stay in the blood, does it reach if you're looking at an infection in the lung, does the compound get to the lung? And then you look at what concentrations in the blood convert to reduction of bacterial load. And that's basically pharmacodynamics. So you need to start understanding pharmacokinetics, pharmacodynamics. And this is, in my opinion, the central point of anti-infective drug discovery. Balancing pharmacokinetics and pharmacodynamics, because with that you start defining the dose, you start defining the dosing regimen, you start defining how many times a day you want to give, so on and so forth. So once an early lead comes through pharmacokinetics and pharmacodynamics, you actually have a refined lead, a robust lead. Now the point why I would, once you have a robust lead, this is a great point to go out and talk to investors, start grant applications. Just remember, you have not solved all the problems. What you have a knowledge base on all the properties of the compound, which then allow you to plan to how to solve the problem. And then once you have a robust lead, you define your CD properties better. you define your TPP better, and that's a great point to go to investors. Okay, then when you come into late discovery, clearly this is more defined as packages. When you go to the regulatory people, what sort of a tox package? It depends on your indication, it depends on the dose, it depends on mechanism of action, it depends on how you're going to give the compound, what patients are you going to go in. All this define your tox package. You need to position the compound in the sense that if my SOC or standard of care is this is penicillin plus or a cephalosporin plus beta-lactamase inhibitor, where is my compound going to fit in? Is it going to go to the resistant types, or is it going to be an alternative? And hopefully you will hear about this in the clinical trials. And lastly, we'll come to the candidate drug where you have the licensing opportunities. I think that's my last slide. Thank you, Nandita. Hopefully I kept myself to time.
Oh, thanks a lot. I think we'll take questions at the end of Dr. Ravi Kumar's talk. So thanks for that, and requesting Dr. Ravi Kumar to make his presentation. Please put your questions in the chat box. We'll take them at the end of this.
One minute, I'll just share. Good morning. Am I audible?
Oh, yes, Dr.
Yes.
Ravi Kumar. Yes. And we can also see your slides.
Yeah. Good morning, everybody. And, I think you had a fantastic session on discovery of the drugs. I think it's a very systematic approach. A talk by Bal Ganesh cannot be different. And he's the one who did a discovery of a drug for TB. So you heard from him. When it comes to discovery of diagnostics, I'm taking a slightly different approach. I don't want to give you a path, though I call it discovery path. I would like to focus more on what should you be internally transforming into if you want to actually discover a new diagnostic. Let me come to this. There are many reasons why people do a new enterprise. Sometimes they just identified an opportunity, or sometimes they're trying to make a difference. In the modern era, they keep saying we want to give back to the society, though it sounds a little bad to me. I'm hitting back at society. And sometimes it's a deep personal experience and an inner calling. But these reasons have to be very clearly thought out. And it's not that because basically these reasons will give a personality to your diagnostic or to whatever you're going to put out as a product. The product will have a personality, and that personality comes from this. So you better watch out for this and actually worry about this. And there are two things. Quite often, if you are creating a completely a new diagnostic, that means with no similar end use, it's not there in the market, you're creating one. There is a totally new path of discovery. I think we will focus more on it. And I'll take you through a few things because I see people coming and presenting in CCamp, in big programs, why they want to do it and how they have arrived at this particular thing. I will take you through that. And sometimes I feel sad. That's not the total picture ever. Now, sometimes you're just improving, replacing, making affordable an existing diagnostic, generally making it faster, cheaper, or easier to use. And those diagnostics, I'm really not going to cover a lot about it, but there your path is damn clear. You have a diagnostic sitting there and you want to better it. You might have a different biomarker, you might have a different, so there is certain amount of novelty and innovation that comes into it, but that's not going to be such a horrendous task as creating a new product. Now, I'll take you through, for example, I'll give you some examples of how we did it. We have defined our why very clearly. The purpose is to save lives, prevent disabilities, and reduce healthcare costs caused by life-threatening infections. Here, if you really focus on it, it looks not lofty, that's not what I am trying to tell you. Really focus on it, our defined customer is actually the patient himself, not the clinician. Though, by the way, drugs and diagnostics, the customer never not the one who orders. He pays for it, but he doesn't order it. The one who orders is it your doctor. And so purposely, I think we consciously entered this and said, this is to save lives, prevent disabilities, and reduce healthcare costs caused by life-threatening infections. And the process is, we said, actionable molecular diagnostics. When we said actionable molecular diagnostics, there are several things that come into play. Can a doctor take a decision based on this and then write a prescription? Even more important, can he alter his prescription based on this? Which meant, for example, we are looking at ICU-based sepsis. For example, if you take a sepsis, we are asking the case, in case there is no organism identified, it's not sepsis and it is something resembling sepsis, is he going to stop the antibiotic? That is the question when we say actionable molecular diagnostic. In the sense of AMR's perspective, that is a very important thing. And the result is we created a syndrome evaluation system called SES, and that's for detection of all pathogens that can cause a syndrome. They could be viruses, bacteria, fungi, or parasites, but all in one sample in a single test, anywhere from 11 organisms to 32 organisms in a single sample. So you just see how the purpose and everything got made the product defined. So we were actually looking, I just wanted to take you through this scenario, how we went through. We said people come with infections to hospitals, they get OPD level antibiotic, 90% get cured, the 10% come back to the hospital. Now, when they come back to the hospital, because they are already on an antibiotic, their cultures are not going to be positive. The regular positivity rate or detection rate is about, at the best in the world, is about overall world statistics. If you take average, works out to be 15% of the people, we know what is happening in the next 5 days in the ICU. The other 85%, we don't know what is the infective agent. So your whole antibiotics and everything gets tuned by this lack of information. Everybody, because, and the second thing is, you're getting, I sent the culture on day 1 and I got the culture on the 4th day. I actually asked the question, how relevant is it today? So I'm still worried about, and I keep giving multiple antibiotics. And this actually led to this situation of 15 to 20 days in ICU for most of the patients on heavy dose of life-saving antibiotics. I'll come to this a little later. I mean, the rest of it. But most important thing is, how do we do these antibiotics? Antibiotics are escalated based on the clinical picture. First day, you may start piptaz and amikacin or ceftriaxone and amikacin. Then second day, you put vancomycin or linezolid. Third day, you start meropenem. Then you start colistin tigecycline, and then you put them on antifungals. And then you start asking, is it a refractory case? And this escalation, people want to stop it by what is called antibiotic stewardship. I think I just want all of you who are entrepreneurs to think, if your kith and kin is in the ICU and you don't know the bug, how much stewardship of antibiotics would you like to be exercised by the clinician? Just think about it. But what you, in fact, we say is, no, that's wrong. Let's have diagnostic stewardship. A diagnostics will tell us exactly what the bug is. Then you will have a fantastic strategy. Well, we are burning the candle from both sides. In OPD, we are exhausting from the lower end of the antibiotics to the upper end. And in ICU, from the top end, we are actually developing resistances. So we are burning the candle both sides. I'm sure all of you heard of this story, but I don't think you have seen it from the ICU viewpoint. Everybody talks about, every lecture I heard is about OPD, how antibiotics are used. Now, I've come to this. So let's say like this, we got an identification of an unmet medical need. This happened in 2007. In 2007, we started and said, sepsis, diagnosis of sepsis is our unmet medical need. Now, then we needed understanding of patient's need. That's what we call as voice of customer, understanding the clinician's need, understanding the lab need. By the way, these three parts I'm coming to are different. And we require a deep dive into the workflow. And wherein the workflow is this diagnostic actually required? Of course, then the data. Let's go and see how people arrive at their TPP or the product profile. A typical interview with intensivist on molecular diagnosis for sepsis. The doctor told me, this happened really in 2006 to 2007. Sometimes I met a set of doctors. Generally, the conversation went like this. I summarized it for you in a slide. I need to know whether it is sepsis or not. Then I say, no, you have sepsis biomarkers like procalcitonin and C-reactive protein. Oh, okay, I want something better sensitivity and specificity. Such a biomarker is not available. What about those cases you're treating as infection? Do you want to know the bug? I ask. Then they say, oh, I want to know it's gram positive or gram negative. That is sufficient. No, I don't want anything more. Then how will you give specific antibiotic with this information? You ask that question. Then he says, no, I don't even want to know the bug. Just tell me the sensitivity. What antibiotic I need to give to this patient? Then you say, no molecular diagnostic can find molecular diagnostics only find resistance, not sensitivity. Oh, then tell me resistance alone. What drugs are not going to work? I don't want to know the bug. Then you ask, what about the bug? For example, resistance gene can be in a commensal organism. A MECA could be in coagulase negative staph on the skin of the patient. And you might get that as a contaminant. And you're detecting that and you're thinking there is staph aureus while he is actually suffering from a gram-negative bacillus in the blood. Then he says, really, can you give species and resistance in the molecular diagnostics? Yes, that is the aim of our work. Well, what is required is what replaces culture and sensitivity. We need everything then suddenly. But I have seen projects being presented in big, saying, I will only identify gram positive and gram negative. Who told you? The clinician told. Have you probed further? Have you asked him further? Have you deep dive into his whole day's life and see how he prescribed? No. The interviews have to be multiple. I want you to do your product profile based on talking to at least 50 to 60 different clinicians doing that work. I know it sounds pretty stiff. Then comes you're making certain early decisions. What organisms? So in sepsis, let's say where data on culture is only 12% of cases, careful extrapolation to the rest 88% is required. We did that. We asked a question, what if some organ, and you will hear from all of them, most of these organisms are not culturable, which is not the truth. Today we know. What antibiotics are used in the ward? What antibiotics are used in ICU? You want treatment algorithms vary from hospital to hospital, and you better find out that. What resistances are seen in the hospitals? What are the genetic markers of these resistances? This was not available in 2008. We used to go and collect resistance colonies, and we actually went into the literature, and let's say if there is an amikacin resistance in Klebsiella, there is a meropenem resistance in Klebsiella, we looked for about 2 to 3 genes. Today we know entire gram-negative bacteria carry about 14 different carbapenem-resistant genes. Today we are looking at that. Similarly, we were at one time point looking for extended spectrum beta-lactamase. We were looking at 3 enzyme genes, TEM, SHIV, and OXA. and their mutations. Up to 40 mutations we were detecting. It's a very complicated molecular diagnostic we made. And you wouldn't believe if I tell you, and all of you would be stunned if I tell you that in 2011, by 2011, we stopped seeing those 3 genes. We started seeing a plasmid acquired gene called CTXM, completely dominating 100% of the colonies. So you need to be worried about, so it's actually a basic work we have done. So sometimes basic research is needed for the diagnostic discovery. You might have to do that. You have to get involved with a hospital, with a microbiology department, with a set of clinicians, and actually ask these questions. I'm just giving my example so that you extrapolate. Who is your target customer? Your TPP depends more on that. Patient, clinician, microbiology lab. Luckily for a drug, this problem is not there. In a drug, you take your clinician as your as your target customer and finish it. This determines the features of your test, and it needs to fit to that workflow of clinician and microbiology lab. You will decide. They're very, very different tests, by the way. If you really look at, there are 2 companies which made multiplexes in 2012, 2014 now. One is Roche, another is a company called BioFire. BioFire is not a multiplex. Roche is a little multiplex. BioFire is massive parallel uniplexes. The whole thing is, one is clinician oriented, another is lab oriented. So most of the labs acquire BioFire because it's the lab oriented. So at the end of the day, how you reach to your customer also depends on what is your focus. Now, in this, quite often, in our case, we went and looked at common genes for bacteria. For example, just to give you an example of how things change, 16S and 23S, and similarly 18S genes, ribosomal RNA genes for fungi, and looked at all of them. We tried 1.5 years, we burned in that process. We knew that you don't get anything specific or anything sensitive. In fact, sensitivity was very low of that. Though it's a multi-copy gene, 16S. So you think you will get enormous sensitivity. No, your sensitivity is compromised by the commensals and contaminants that come while you're taking a blood sample from the patient. This was not envisaged by a lot of people. And a lot of tests have failed US FDA because they keep detecting commensals again and again, just like the culture. So in this, where you have to make these decisions, you have to worry about a little deep dive into the science and the logistics of how you're going to, what is your chance? Logistics means not some external logic. What are your chances of finding these 2 primers actually going and getting your pathogens DNA amplified? You need to look at that possibility. And we have done in silico a lot of work, but most of it, about 70% of it, was never proven in actually when we did experimental. In a wet science, 70% of the time we had to vary things. Why am I giving about all this is, many of people think, I have a set of primers, so I have a PCR. I have a set of primers and a probe, so I have an RT-PCR. No, it doesn't work like that. At the end of the day, it is sufficient to say when you have a multiplex, we have 10 organism to 12 organism multiplexes, which worked very well. Those set of genes and primers and probes that worked very well. But we discarded them because we wanted totally 20 different organisms in the sepsis panel. We just discarded all of them and rewent and completely reworked out everything. It's a trial and error process. Every diagnostic, I want you to understand it needs to be tested in the clinic. All the time, you need to bother about how is it working on a patient sample. You have to be integrated, well knit with a hospital for you to do that. It's very, very important.
Dr. Ravi Kumar, another maybe 3-4 minutes.
No, I think I can even stop here and invite questions so that we can have more discussion. I think I made a point, but I hope I got through.
Okay, sure. We'll take questions. Dr. Ganesh. Dr. Bal, there's a question to Dr. Bal Ganesh.
I'm here.
Yeah, sir, can you please brief about the grants available for drug discovery? Just to say that we will be covering this later, but if you wish to take the question.
I think it's better covered as a whole rather than me just saying. If you just look up BIRAC, you will see that right from discovery, early discovery to early lead to taking the lead forward, there's a whole lot of grants which are competitive and available. Then, of course, you have the CARBAX, which I think is more related to infection. Let me also tell you that if you have a good lead, that you will find the grant which take you up. It's a question of finding a good lead. I won't go into the various grants simply because, Nandita, you guys are going to discuss this in more detail.
Yes. Thank you. So there was a question which was answered by another participant.
Yeah,
what is FOR, frequency of resistance?
Right.
So that was the thing. The other question is, Dr. Bal Ganesh, sir, excellent talk. My question is, drug discovery initiative, I understand, is also impeded by, sorry, I'm not able to read this.
I think it's talking about return of investment, how initiative can be made lucrative,
Yeah.
which would result in hastening. Yeah, this is a huge issue. Many factors are contributing to the fact that infection is no longer, or a new anti-infective, and infection as an area is not lucrative. I think the wheel will come around. Lots of initiatives now, which what you call as pull and push initiatives, incentives coming both in the US and in Europe, where hopefully this will help in this whole issue of return of investment, of how antibiotics, how we are going to get antibiotics reimbursed, and how companies can get at least some a guarantee on their investment that they put up. So there's a lot of things happening. It's a separate topic by itself. I don't know, Nandita, whether you have it.
We are covering it towards the end.
Please do. I think that's extremely important. But yes, point taken that there is an issue with the return of investment. I think it's important to know that a lot of things which are happening to help.
Thank you. Dr. Ravi Kumar, a question to you. Even if we identify the bug, how sure are we that the antibiotics will work? Because the bug is evolving.
Yeah. It's an interesting question, but there are two aspects to it. First, if you don't know the bug, there is no way that you can actually ascribe a resistance or a sensitivity to that, because there are multiple genes which can be in any single bug causing resistances. So you better know the bug and then match the resistance. In a diagnostic, always focus on that. Simplest example I gave cons. There are sometimes the contamination because of the improper taking of the thing, so many other gram negatives which are not sepsis causing organisms. Simple. Gut E. coli containing carbapenem means nothing. means nothing because it's not an invasive E. coli. It doesn't come in and doesn't cause a sepsis. So you need to know the bug. The most important thing is all the time you need to be worried about what resistances. Let's say if you're working on Klebsiella, you need to be taking all through. Last 10 years, we haven't stopped this work. We keep taking samples from various hospitals, Klebsiellas, and then ask the question, are we able to match with our genotype their phenotype? If not, we are on a new investigation. You need to be worried and all the time ahead of it. It's like the game of creating viruses in the computer, and hackers keep creating, and we keep creating antivirus programs. This game will go on with the bugs.
Thank you, Dr. Ravi Kumar. Just before I take the next question, I think Dr. Bal Ganesh has another meeting and needs to leave. So just would like to thank him for his, spending his time and really trying to cover. I know that it's a really huge, expansive topic, but I think for this session, he's been able, thanks a lot for covering it and all at least the important aspects. And I think we will look, if there is a need to get, into deeper depth, we will do, shorter sessions or rather longer sessions on, on focused topics. So we will come back to you again. But thank you so much, and look forward to staying in touch.
Thanks, Nandita. Thank you, everybody. Hope to see you guys again. Bye.
Yeah, bye. Thanks. Yeah.
Bye, Ravi.
Bye.
Then there's another question. Carbapenems hydrolyze specific carbapenems. What about the AMPC and ESBL genes hydrolyzing activities?
I didn't get the question completely. I was reading that question. Let me answer that in a slightly different way. AMPC and ESBL genes have to be looked into in gram-negative, especially Enterobacteriaceae. It's very important because they can cause a set of resistance. It may not be carbapenem resistance. ESBLs, by definition, ESBL is not carbapenemase, but Europeans do include carbapenems under ESBL. But I am trying to make a distinction between extended spectrum beta-lactamases, which mainly look at cephalosporins, versus I'm looking at carbapenemases. When you look at ESBLs in this, if they are not there, and let's say carbapenemase is not there, you are actually in a fantastic situation to treat that case. You can cut down your antibiotic level to a minimum. So that's why you need to know them. I know that there are a lot of people trying to look at AMPC, and we do look at ESBLs. Now I told you the story of ESBL 3 enzymes we were looking. Now we don't find them, and more than 98% of all these are only CTXM based plasmids. So ESBLs have, but the situation will change. That's what I believe.
So very nice, sir. How 16 sRNA detection is useful in gut microbiome? Yeah.
See, in a research, 16S RNA or 23S RNA is very different when you're using it for research. It's a very powerful tool. You're doing sequencing. You're actually ascribing the species. That's wonderful. But if you want it as a diagnostic for your critical care or for, let's say, urinary tract infection or so, you are going to have problems with it. That's what I mean. It's a great research tool, no questions asked. So in gut microbiome, it's a wonderful tool. You go completely sequence the 16S ribosomal RNA, the DNA, and then you get your sequence, and it will also give you the species and subspecies and strain differences. A lot of things can be done with that 16S in a research level.
I think there are other questions which will be answered in subsequent sections regarding funding and how do you get things to the market. Clinical trials is the next one. Dr. Kher is already here with us. What is the future? Just one last question. What is the future of future prospects, I would say, of plant-derived antimicrobial compounds for treating resistant bugs in terms of safety and efficacy?
Field is wide open. In fact, there are wonderful projects in Big itself. I have seen about 2 or 3 of them. They're actually based on unusual properties of the bacteria and the antibacterial activities of known spices. I remember somebody trying to dislodge biofilm forming bacteria using antiquorum sensing molecules from cloves. Things like that, there is a great possibility. But one thing I can tell you, whether it is plant derived, whether it is synthetic, or whether it is a fungus derived, your antibiotic discovery journey is the same. The same rigor of toxicology, the same rigor of its teratogenicity, the same level of stringency has to be applied. So it might give you a great lead in the sense the target might be easy to identify in a plant-based drug if you have already got the fractionation. If it is a total plant product which is giving you safety, I advise not. Please don't start up a company based on that. The hunt for that molecule in that mess is something which many of us are not capable financially. Nobody will support you a fishing expedition.
Thank you. Thanks a lot, Dr. Ravi Kumar. So, of course, as usual, always a delight to hear you. And so thanks a lot. I think your experience in in the diagnostic space is absolutely amazing, and I think has also in many ways like especially the last sentence that you mentioned, right? Like we are so sometimes tied up with our own research, but I think one has to look at the as I say, the ROI, or whether there is anything in, if you want to do it as a venture. I think to continue research, it may be fine. So I think very, very valuable insights, not just on the science, but even for entrepreneurs as well. Thank you so much for coming and speaking with us.
If you don't mind, I'll just make one statement.
Please. Yeah.
Sometime back, Roche has floated a company to look for natural compounds from sea animals and seaweeds and so on. Roche spent more than $3 billion. They landed with number of plant mixtures, not a single compound. That is an example of why natural compound research is very tough to do. That's all. Thank you.
Yeah, thank you. Thanks, Dr.